Today we have the pleasure of having with us Professor Jane Skok. She's originally from South Africa, but she did most part of her important work and stayed for a long time in UK. And she did there her PhD, University College of London, in immunology and genetics, the postdoctoral training with Dr. Amanda Fisher and David Gray in the Imperial College School of Medicine. And from there, he moved to the NYU University, New York University, when she got promoted from lecturer, assistant professor, associate professor. And she is now an associate professor at the university. She has, she's also, they mentioned this one, but now she's the Sandra and Edward Meyer Professor at the Department of Pathology. Her work has always been devoted to understanding the epigenetic regulation of lymphocyte development through chromosome dynamics and nuclear architecture. And she has done seminal contribution to understand the architecture of chromatin, mostly in B cells, but also in other immune cells. She has received many recognitions by her work, by academia and foundations. So we're very happy to have her here. And the title of her talk is Architectural Proteins and the Role in Chromatin Organization and Gene Regulation, something a little bit similar to this title. So a great pleasure, Jane.
Okay, thanks so much for inviting me here. It's great to be here. This is one of my favorite cities in the world. And I'm hoping that I can stay and enjoy a bit of it for the next few days. So I'm going to start. The title of the talk is about CTCF mutations that are found in cancer and CTCFL in cancer and their role in altering chromatin architecture and gene regulation. So I'm going to start by acknowledging the people who did the work. So a lot of wet lab people are involved in this project. And a lot of people sort of transitioned through this project because of COVID. It started with Nishana, and she was joined by Priscilla and has help from Boris and Faye and Gueme. And the bioinformatic people on this project are Catherine Doe and Theo and Ricky Agosh, who did some biophysical modeling for us. So before I start, I'd just like to say, if you want to interrupt, if you have a question, please do so. It's nice to get feedback in the middle of the talk. So we know from imaging technologies that chromosomes occupy these discrete territories in the nucleus. And these discrete territories can be broken up into active and inactive chromatin. So the active chromatin is shown here in the blue, and it's more pointing in towards the center of the nucleus. And the inactive chromatin is more, is the orange kind of colored chromatin, and this is more associated with the nuclear lamina. And these territories can be subdivided into compartments, which are A compartments and B compartments, which are active and inactive compartments. And these can be further subdivided into what is known as highly self-interacting topologically associated domains. And these are separated from each other by insulated boundaries made up of CTCF and cohesin that bind at these boundaries and are enriched there. And it's known that CTCF plays a key role in organizing chromatin into TAD structures by promoting the formation of loops and boundaries that are important for gene regulation. And cohesin rings basically create loops by actively extruding the DNA. So they jump on the DNA and slide down the DNA until they come into contact with two CTCF proteins that are bound that are in a head-to-head orientation. And this was nicely shown originally by modeling and also more recently by Jan Michael Peters' lab, who showed this in an in vitro system. So the function of TADs is thought to be that to restrict the range of enhancer actions so that enhancers can really only control the genes that are within the TAD that they're located in. And if you mutate CTCF, and you lose the boundary at TAD, you can see that genes that were once outside of the TAD can come in and now be controlled by enhancers that didn't normally control them. So this can really mess up gene regulation. And a really nice example of this is one from Danny Reinberg's lab that we collaborated on. And this is looking at the HOX-A structure. And in motor neurons, what you see is that the HOX-A structure is, the HOX-A is divided into these rostral and caudal domains, and the active region is in the rostral domain, and the inactive region is in the caudal domain. And these are separated by a CTCF site here. And so the Reinberg lab deleted the CTCF site. And what you can see is that in normal wild-type cells, if you put a viewpoint here and do a 4C-seq an analysis of chromatin interactions, you can see that all the interactions are in the rostral domain, and these are not interacting with any of the regions in the caudal domain. But if you take out the CTCF, now you can see that this interaction spread a bit into the caudal domain, and now you're incorporating the HOXA7 gene into this domain, which it wasn't there before. And this is linked to a diminishment of H3K27 trimethyl, a repressive mark, and increase of H3K4 trimethyl in this region, and RNA pol2. So CTCF site removal can cause spreading of H3K4 trimethyl to the HOXA7 gene and lead to its expression upon differentiation. And deletion of CTCF at the site also results in loss of a rib and homeotic transformation in mice. So we know from these and many other studies that disruption of a CTCF binding site is sufficient to alter 3D chromatin structure in a manner that leads to alterations in interactions and spreading of active histone marks and changes in gene regulation. But what we don't know is whether CTCF alterations can actually drive oncogenic transcriptional programs. So that's what we set out to answer. And you can see here that CTCF mutations are found in many different types of cancer, but it's really not known what impact these mutations have. So CTCF is an 11 zinc finger protein. It has a large N and C terminal globular domain, and the zinc fingers 4 to 7 make contact with the DNA at this consensus 12 to 15 base pair consensus binding site. And we and others showed that the N-terminal domain is important for interacting with cohesin. And this explains the requirement for the convergent CTCF binding sites in stopping the movement of cohesin on chromatin. And we also showed that the N-terminal and zinc finger domain are required for CTCF's insulating function. So to study mutations, we decided to select those that occur most frequently in patients. And also we selected them based on the location of where they were in the zinc fingers. So we're only studying mutations in the zinc fingers at this point. And there are different types of mutations that you can see in the zinc fingers. The zinc coordinating residues are marked here in red. And generally it's thought that if you mutate any of these, it will lead to loss of binding of the CTCF protein. And then there are these residues that make direct contact with the helix of the DNA at -1, 2, 3, and 6, and these are shown here in pink. So these are important. And also there are these residues at -5 and in the linker region, and these make contact with the DNA. These have phosphate contacts with the DNA. And so these are important in terms of charge, and they also are important for stability. And then there are these boundary residues at position -2 and 9, and these are important for interactions between the zinc fingers. So they are important for stabilizing the protein, and also for that's going to impact binding. So these are the mutations that are highlighted here that we decided to study. You can see the frequency in the number of patients, and also you can see the location in the zinc fingers here. And this is the sequence of the difference to which the different zinc fingers bind. And there was a crystal structure that came out a while ago from Jiadong Cheng's lab in 2017. And he'd looked at the crystal structure of the zinc fingers 3 to 7. And now he's got an additional new crystal structure, which incorporates zinc finger 1. And we've been collaborating with him on these mutations to look at the impact of what happens with the mutations to the binding and the structure. And here you can see the different mutations that we're studying and the location on the zinc fingers and the contacts that they make with the DNA. So our hypothesis starting out, going into this study was that each CTCF mutation will alter cellular function in a different way, depending on whether it's associated with either total loss of CTCF binding, and we expect to see this in mutations in residues that coordinate the zinc, or a change in binding affinity stability, and this could be potentially any mutated residue, and then an alteration in binding motif preference. And we expect to see this in residues that make direct contact with the alpha helix of the DNA. If these are mutated, we could expect to see a different preference for binding motif. So the system we used to study this was a system that was developed by Alphege Nora and Benoit Bruno, and it's an ESC auxin-inducible Degron system, which has an AID tag in the endogenous CTCF locus and an EGFP so you can follow expression. And we inserted into this a DOX-inducible transgenic wild type or mutant CTCF proteins, and we put them in the tiger locus. And these have a flag tag, so we can distinguish between these and the endogenous CTCF. And they also have an mRuby fluorescent marker, so you can follow them by fluorescence. And basically, the way that we do this analysis is that we select transgenic clones that have the same levels of expression for comparison. So we have a wild type transgenic clone and a mutant transgenic clone. And the reason that we do this is because the transgenic clones don't express at the same level as the endogenous. So we can look at expression of these mutant CTCFs in the presence or absence of endogenous CTCF. And we like to look at them in the presence and absence because in the presence of CTCF, endogenous CTCF, that's what you would normally see in a patient because only one allele would have the mutation. So this is typically what we see for the mutants. This is the K365T mutant. It's a mutant that makes, in a residue that makes specific contact with the DNA, and it's in zinc finger 4. And what we typically find is that we lose a lot of binding sites, and there are common binding sites, and we gain de novo binding sites. So here on the top, you can see in this panel, the profile for the wild type CTCF sites only, the common sites and the mutant only sites. And most of the time, what you see is that the common peaks are occurring at the strongest CTCF peaks. So the green is the wild type and the red is the mutant. And here we're looking at the top is the binding profile in the absence of endogenous CTCF. And this is the binding profile in the presence of endogenous CTCF. And this is very nice for looking at a binding affinity stability, because here you're looking at a competition of between the transgenic wild type and mutant CTCF's ability to compete with endogenous CTCF. And this kind of reflects how much stability is. So you can see here that the mutant is really not so able to compete with endogenous compared to the wild type, and there's a much lower peak here. So we use a pipeline called Cis-Diversity, which was published recently in Genome Research, to look at binding motifs where these mutants will bind. And this is a very kind of unbiased way of looking at this. And what I'll explain how this works. So this is the wild type only peaks. These are the common peaks. These are the mutant only peaks. And here on the bottom, you can see the consensus motif in every case within the red box. And then these motifs 1 to 4 are the different motifs that we see for each set of profiles. And 1 is the most frequent binding site, all the way down to 4, which is the least frequent binding motif. And what we see is that in the wild type only peaks, you have the consensus motif. But you also have the consensus motif most frequently occurring in the common sites. And where you see a difference is in the de novo binding sites for the mutants. And here you can see for this mutation that occurs in zinc finger 4, you normally have a GGC triplet that this zinc finger binds to, but now you can see that it's altered to a GTC. So this is the zinc. This is the crystal structure that was that hasn't been published yet, and this just shows you the contact that the K365 is making with G11. And you can see that in the wild type case, you have this bond here that is making contact with G11, but when you mutate this lysine to a threonine, the hydroxyl side chain really can't form a hydrogen bond with guanine. So that kind of explains why you have less stability of binding. And here you can see that if you look at binding of, this is like turning this around now to look at the binding site that the wild type will bind to, and the bases that it prefers to bind to. The lysine 365 prefers to bind to a G, and T is less stable in the green, and you can see the A is even less stable, and the C is even less stable. And this can be explained because the lysine has 2 hydrogen bond donors, and G has 2 hydrogen bond acceptors, so this is the strongest interaction. The T has 1 hydrogen bond acceptor, so the charge is less negative than the G, and A has only 1 hydrogen bond acceptor, and so this is kind of a neutral charge, and C has no hydrogen bond acceptor, so that's why you see this, that this is the weakest interaction. But if you look at the mutant now, you can see that the mutant doesn't really care where it binds. It binds to pretty much all of these bases with equivalent stability. So here's another mutation, the Q418R. This is in zinc finger 8 as zinc finger 6, and this again is in a residue that makes specific contact with the DNA. And again, you can see here the profile is most strong at the common binding sites, and you can see here that this mutant is really not, is very weakly stable compared to the wild type when you look at the competition with endogenous CTCF. And again, we can see the same story here, loss and gain of binding, and at the wild type only peaks, again, we see this consensus CTCF motif coming up most frequently, and same for the common sites, but again, you find a new motif for the de novo sites, and this is changed at the site where the mutation is occurring in zinc finger 6. So this A base that Q418 binds to is an interesting base, because if you look at hotspot binding site mutations in cancer, and particularly in colorectal and gastric cancers, where you see 25% of these cancers and 19% of colorectal cancers have hotspot mutations in CTCF binding sites. And you can see that one of the most highly mutated residues is this A base in the middle. And actually, if you, at about 3,000 binding sites in the genome have this site as a palindrome. And if you mutate this A to a different base, you can change the orientation of CTCF binding, which can screw up the regulation of genes in the neighborhood. So this is another mutation. This is in a phosphate contact residue here. And you can see that there's, again, you lose a binding stability when you look at the common sites, a lot of gain and loss of binding with the mutant. And here, same story, that you have this consensus motif coming up in the wild type only sites, in the common sites, and in the sites where the mutant binds de novo, we now see the most common motif is this simple repeat. So this is an interesting mutation because it's in the zinc finger 7, it's the R448Q. It's again a residue that makes contact with the DNA. And now you can see that this mutation actually causes a more stable binding than the wild type. So it's better able to compete with the endogenous compared to the wild type. So same story again, consensus motif at the wild type only sites and at the common sites, but then there's a change of sequence for zinc finger 7, and it preferentially binds to TCA rather than CCA. So this is a mutation in the zinc and a residue that's coordinating the zinc. And you can see that we really do lose most binding sites here. And this is the only mutation where we see that the stronger sites are in the wild type only peaks. But you can see that where there's the few binding sites that they can bind together, it's really much less stable than the wild type. And again, same story, consensus motifs at wild type and common sites, but now in the de novo sites, you get this simple repeat. And so the question that we wanted to know is, what's the impact of these mutations on cohesin localization? So I'm showing you a few examples of these 3 mutants here. And these are looking at the proportion of wild type CTCF that overlaps with cohesin at common binding sites. So for each mutant, this is a different set of common sites. So you can see in the K365T in the wild type situation, cohesin overlaps with CTCF at 59% of sites. For this mutant, 53%, and for this mutant, 51%. So similar level, but when you look at what happens when the mutant is binding, what you can see is that for this mutant, you really lose a lot of cohesin at the site. This is less cohesin lost at these sites for this R377H mutant. And for this mutant, which is the mutant that binds more stably than wild type, you really don't see a change in cohesin binding. So this is also now looking at the wild type only sites. Oh no, so this just shows you the level, the number of mutation sites that are affected in each case for the common binding sites. And this is looking at wild type only sites. So what you see here is that these wild type only sites are much less able to stop cohesin binding than compared to the common sites. And this is the number of sites that we see in each case. And then looking at the mutant only de novo sites for each mutant, you can see that these sites are really not so efficient at stopping cohesin movement on chromatin. And the most effective one is the R448Q mutation. And that's the most stable mutant that we have that's able to compete with wild type CTCF. So basically, just summarize this part of the talk, I've shown you that in general, common binding sites are the strongest CTCF binding sites, and the most predominant motifs at wild type and common sites are consensus or similar to consensus sequences. And cohesin overlaps less frequently at wild type only sites compared to the common sites, although the motifs are actually pretty similar. And this suggests that at these sites, maybe that the common sites, maybe other factors are contributing to the insulation effect. So we wanted to know what the impact of CTCF mutations are on gene expression. And basically what we see is the severity of gene expression changes in mutants is linked to the severity of changes in the binding profiles and binding stability of the mutants. So here I'm showing you different mutations, and here I'm showing you the least stable binding, and this mutant has lost and gained the most binding sites. And at the bottom, just showing you the competition with endogenous CTCF. And so when you compare between mutants, be aware that there's differences in scale here. So you can see this mutant, which is the Q418R mutant, it has the most changes in gene expression, and these just go down in number depending on the stability and changes of loss and gain of binding sites. So you see the least changes are happening at this mutant, the R448Q mutant, which is the most stably binding mutant. So we wondered whether CTCF mutations could affect the accessibility of transcription factor binding sites. So we looked using a Tobias pipeline at transcription factor binding and predicted differentially bound transcription factors using ATAC-seq footprints in the wild type versus the mutant. And here you can see that in the mutant, these are all the transcription factors that are predicted to bind more strongly in the mutant, and these in the wild type. And you can see that the CTCF and CTCFL are coming up in the wild type. which is sort of a confirmation that this is working. And this is a different mutation, the K365T. Again, transcription factors that are found, predicted to be found more often in the mutant, and these are the ones that are predicted to be found more often in the wild type. And now what we've done here is look, actually this is the same mutant that I showed you before. And now what we did is to look at differentially expressed genes and associated promoters with predicted differentially bound transcription factors in wild type versus mutant cells. And you can see that enrichment of differentially expressed genes and associated promoters with predicted differential transcription factor binding is very significant in the wild type and in the mutant case. So this is showing you the same thing for the R377H mutant. And this is again showing you the overlap of differentially expressed genes and promoters with predicted differentially bound transcription factors. And you can see here for the R448Q mutation, which is the stable mutation, you see much less changes in predicted transcription factor binding in the mutant. So, and again, less changes of gene expression that are overlapped with these changes. So what effect does each mutation have on CTCF protein structure and its association with DNA? Well, we teamed up with Tamar Schlick at NYU and Stephanie Portillo in her lab and Rikia Ghosh, who's in her lab, to do some biophysical simulations of CTCF DNA wild type and mutant complexes to determine how each mutant affects the protein structure and its association with DNA. So this is a different crystal structure. This is the crystal structure of zinc fingers 3 to 8 that was published in 2017. And you can see the wild type and how the zinc fingers all make contact with the DNA. And now if you look at all the mutants, and the mutations are highlighted, the location of the mutations are highlighted by an arrow in each case, you can see that the zinc fingers binding is altered. And this impacts not only the zinc finger that the mutation occurs in, it also impacts the binding of other zinc fingers, which likely is going to affect the stability of binding of the whole protein. So next we asked, what is the impact of CTCF mutations on chromatin organization? So we did high C. Here we're looking at the K365T mutation. This is a heat map of interactions and on the high C profile for the wild type and the mutant. And this is a differential contact map showing the changes in binding, changes in contact between the mutant and wild type. And you can see by the red that there's more increases in contact in the mutant situation here. So we're looking at this integrin ITGB1 gene, and the location of it is shown on the map here as the red bar. And here you can see the changes in contact frequency are linked to changes in the binding of the mutant. This is the wild type transgene compared to the mutant transgene. So you can see that there are additional binding sites in the mutant, and some of these are altered in binding the height of the peak, but you also lose some of these peaks along here. But when you look at the cohesin, this is the wild type cohesin, and this is the mutant cohesin. You can see that the cohesin is pretty much lost in this region, even though there are binding of the mutant CTCF. And so this is linked to changes in expression. So you have nice gene expression in the wild type, and this is pretty much lost in the mutant. And this is another example. This is the DNMT3A gene. Here we're seeing a lot of loss of interaction. And this loss of interaction can be connected to, again, changes in CTCF binding. So there's peaks here, and there are much smaller peaks here, and you lose a peak here. And you can see that this is the cohesin track, and this is the mutant cohesin track. And pretty much you don't see much of the peaks that you see in wild type. And again, DNMT3A expression, which is expressed nicely in the wild type, is very much reduced in the mutant. So here is the Hoxsey cluster, which is where we started off. And this is just showing you the effect of different mutations on contacts within this locus. And on the top here, you can see gain on the top and loss of loops in each case. And mostly you're seeing loss of loops in all of the cases of mutant, but you also can see that there are differences between each mutation. So this is the K365T mutant. You get this is the Hox cluster here, which is shown here in this very small subtat. It's a mini TAD actually, and you can see the location of where we are here. You can see that there's a lot of loss of interaction in this TAD, but gain of interaction on either side of the TAD, in the TADs, the neighboring TADs. And a similar picture for the 377 mutation, but less strong effects. And this Q418R has even a completely different profile altogether. Loss of interaction again in the Hox, but different gain of interaction in the TADs on either side. So this again is linked to changes in binding profile of the wild type of mutant. So now this is slightly differently portrayed. Here we have wild type of mutant CTCF and wild type of mutant cohesin. So the wild type's on the top and the cohesin's on the bottom. And you can see changes in expression in each case. And the changes in expression and binding profiles are different for each mutant. So it has a different impact in the locus. And this is another example. This is the MYC locus. You can see that mostly you get loss of interactions, a slight gain of interaction in the Q418R. And this is the high C profile differential interactions in each case. And again, you can see here that in the wild type versus the three mutants, there's this nice CTCF peak at this location. It's enriched at the R44AQ, which is the stable mutant, and it's completely lost in the Q418R. And this is linked to a gain of MYC expression in this Q418R mutant. And there are also changes of CTCF binding in the PVT locus, and you mostly see a gain of binding in this location, and the gene is slightly changed in expression. So one more example here, which is a locus where you have differential interactions in wild type and mutant, and you can see the high C maps in each case. But here now, even though there are changes in CTCF binding and cohesin binding, there is really no change in expression. So there are some places where you get these changes and you don't see an impact on the function. So the mutations in CTCF are linked to cancer as shown by gene set enrichment analysis. This is the profile for each of these four mutants. And you can see there's some things that are similar and some that are distinct for each mutant. So to summarize what I've told you so far, all CTCF mutations bind a subset of CTCF binding sites, as well as de novo sites. And with the exception of the H455R mutant, which is the ZIC coordinating mutant, the common binding sites are always the strongest CTCF binding sites. And these have consensus motifs. And these mutants can bind with lower and higher affinity at these sites, depending on the mutation. And the mutants differentially affect the ability of CTCF to block the movement of cohesin on chromatin. And all mutants have an impact on gene expression, transcription factor binding site accessibility, and chromatin interactions. And this is linked to their individual binding profiles and stability of binding. And there are examples where all mutants affect a particular locus in a different way. And there are many of these genes. And there are others that are altered that are linked to cancer, as shown by the gene set enrichment analysis. So this provides us with both a loss of function and gain of function model to test the impact of de novo CTCF binding at numerous sites on cohesin binding, TAT structures, and gene regulation. And it's actually a very nice resource for looking at whether your favorite gene is actually impacted by regulation of, by the binding of CTCF and cohesin at around the locus. So we are currently making mouse models to determine whether mutations can act as drivers of cancer or whether they have an impact on the prognosis of particular cancers. And we're doing this in collaboration with Diana Saifontis and Ari Malnik. So I'm going to move on and switch gears now to discuss CTCFL. This is some published work that I'm going to talk about at the beginning and then go on to some new work that we've been doing that's not published. So the original published work was done by Nishana, and she was working on this project defining the relative and combined contributions of CTCF and CTCFL to genomic regulation and their impact on cancer. And basically, CTCFL is also known as Boris, arose as a result of a duplication and transposition event. It's a paralog of CTCF. It has 11 zinc fingers, just like CTCF, but its N and C-terminal regions are very different. And you can see the differences in the residues between CTCF and CTCFL here, shown in yellow. So a lot of the residues in the core zinc fingers that make contact with the consensus binding site for CTCF are very similar, as you can see here. And so CTCFL is transly expressed in premeiotic male germ cells together with CTCF, and is thought to play a unique role in spermatogenesis by regulating expression of testis-specific genes. And in this context, it functions as a transcription factor. And it's amplified and mutated in many different cancers, as you can see here. And I'm highlighting melanoma and ovarian cancer here because this is the context that we study this in. So you can see the expression level by RNA-seq in the different cancers. And again, you can see that in melanoma and ovarian cancer, levels are way above what they normally are, which is pretty much thought to be nothing in normal cells. So CTCFL binds competitively to a subset of sites that CTCF binds, as well as a unique set of sites. And when it binds competitively, it kicks CTCF off these sites. But it's not understood to what extent CTCFL deregulates transcription on its own by acting as a transcription factor or by opposing CTCF. So that's the question we set out to answer. And this is a system that we use. It's the same system that I described before for the CTCF mutations, but instead of putting a CTCF mutant in the tiger locus, now we have put the CTCFL gene into this locus. And we flag tag it because there are no decent antibodies for detecting CTCFL. And we analyze the impact of CTCFL in the absence and presence of CTCF. And in cancer, what you would normally find is that you would have the two together present. So when you express both together, you can see that there are a lot of changes in gene expression in the mouse ES cells. And you can see here that CTCF has its own binding sites, and there are common binding sites for CTCF and CTCFL, and sites for CTCFL only. So this is CTCFL binding in the presence of endogenous CTCF and in the absence of endogenous CTCF. And you can also see there's slightly different motifs for the CTCF only sites. The CTCF seems to have a preference for this A base here, which is lost when in the CTCFL only binding sites. And the CTCFL binding sites have a preference for the C, which is much less required for binding of zinc finger 4 in the CTCF only sites. So we also see that CTCF only binding sites are predominantly intronic and intergenic, whereas CTCFL only binding sites are mostly at promoters in line with its action as a transcription factor. And the common sites are somewhere in between. So there are also site-specific differences in boundary insulation scores at the different CTCF only, common, and CTCFL only sites. You see most insulation occurring at the CTCF only sites, which is what we would predict, and the least at the CTCFL only sites. And you can see that cohesin overlaps with the binding of CTCF, and this is CTCFL expression in the presence of CTCF. And when you take out CTCF, you can see that all the cohesin has pretty much disappeared. So CTCFL is unable to stop the movement of cohesin on chromatin. And we showed that by co-IP that we could, with a RAD21 antibody, which is RAD21 is a part of the complex of a component of the cohesin complex, that we can pull down RAD21 with CTCF, but not with CTCFL. And then this is showing you the effect of CTCFL on chromosome structure. So this is in the uninduced condition. You can see a nice profile of TAD structure. If you take out CTCF, you completely lose the TAD structure. If you rescue with CTCF, you can rescue the structure. But if you rescue with CTCFL, the structure doesn't come back. If you look at what happens when you express CTCF in the presence of endogenous CTCF, you can see a stronger profile of interactions. But if you express CTCFL in the presence of endogenous CTCF, you're losing the interactions that you see when you don't have CTCFL present. And you see a similar thing for loop aggregates. So here is the loop aggregate in the uninduced condition. You take out CTCF, you lose the loops. If you put back wild type CTCF, you can see that you regain the loops. If you put back CTCFL, you don't go back to wild type levels. Sorry, this is, you don't regain the loops. And when you express CTCFL in the presence of endogenous CTCF, you're kind of weakening the loops. So we went on and made a few chimeras, and I'm not going to go into the details of all of these, just to show you one of these, where we replaced the CTCFL N-terminal region with the CTCF N-terminal region. And when we did this, we could again rescue the interaction of CTCFL with RAD21. So it seems that the N-terminal region is the region that is responsible for this interaction. And this has been more precisely pinpointed by other labs. So here, if you use this chimera, you can see that this is the wild type CTCF interaction profile. If you take out CTCF, you lose it. If you put back the CTCF chimera, the CTCF-CTCFL chimera, you partially rescue the TAD structure. And you can also partially rescue the lost insulation that you see when you take out CTCF, when you have this chimera. So what we showed is that the N and C terminal and zinc finger domains of both CTCF and CTCFL contribute to the specificity in how each paralog targets DNA and regulates gene transcription. And I didn't go through all this, but it's published. And we show partial rescue of insulation mediated by the N-terminal and zinc fingers of CTCF. And chimeric proteins demonstrate that the N-terminal domain of CTCF mediates the interaction with cohesin, which explains why you have this requirement for convergently bound CTCF in stopping cohesin movement on chromatin. So we just moved on from studying the mouse to the human CTCFL. And human CTCFL is very different to the mouse because the mouse has one isoform and the human has been reported to have 23 isoforms that are alternately spliced and predicted to generate 17 different protein structures. So this work has been done by Gueme and Catherine. So the original paper identified these using 3' RLM race, and they did this using testis and K562 cells. So testis is where you normally see expression of CTCFL. So we wanted to go back, so this is again showing you expression levels in the melanoma and ovarian cancer cells. But we wanted to go back and look with more modern techniques to see if we could identify these isoforms. Because nobody, when they look at CTCFL in cancer, distinguishes between any of the isoforms. And nobody has looked even to see what isoforms are expressed in particular cancers. So we started by doing some, so you can't use RNA-seq to identify isoforms because the reads are too short and the similarity between the different isoforms is too great. So we decided to do some PacBio Iso-seq, which are long reads to identify these isoforms. And the company assured us this would be possible, but basically there were insufficient reads when we did a genome-wide analysis. So then they said, do some targeted sequencing. So we did targeted PacBio, and we did pick up more isoforms, but you'll see that there's a problem even with this. So these are the isoforms that we pick up with the targeted Iso-seq, and this is in melanocytes, healthy melanocytes, which are the negative control. melanoma cancer cell lines, metastatic melanoma patient short-term cultures, and this is a positive control, human testis, and this is the an ovarian cancer cell line. So basically, we wanted to then validate these by PCR, and this is kind of difficult because the sequence, there's many sequence overlaps between the isoforms, but I have a very resilient postdoc who persisted with this, and she designed lots of primers for each isoform and then performed PCR sequencing to check what's present. So here's what we see in ovarian cancer cells. These are the different isoforms. You can see we identify 7 isoforms that are known and a new isoform that was not known before. And you can see that in the melanoma cells, we only identify 4 of these 8 isoforms. So when she goes to, when Guema went to validate these, what she found is that all the isoforms that are present in the ovarian cancer cells are also present in the melanoma cells. They're less enriched, but they're definitely there. So the validation picks up isoforms that were only detected in the ovarian cancer cells in the melanoma cells. And we could see that both of the melanoma and ovarian cancer cells have this new isoform, C85. And this just highlights the shortcomings of Iso-seq. There are not enough reads to detect all isoforms. And so when we find an isoform, we know it's there, but we don't know what's missing. And we don't know and it's not quantitative either. So she went on to look at a lot of different cells to validate expression of these and look at their profiles. So these are healthy melanocytes, primary melanoma cell lines, metastatic melanoma, and metastatic patient short-term cultures, and also the positive control, which is the testis, and the ovarian cancers, which seem to have the highest expression of CTCFL. So basically what she sees is that you can see that these are all the isoforms down here. These are the panel of cells along here. And you can see that expression of each isoform is very variable depending on the cells. And you can also see that something very interesting, which is this C90 isoform is expressed in healthy melanocytes. And these were not predicted to have, healthy cells are not supposed to express CTCFL. And you can also see that in the metastatic cells, expression of the isoforms is higher than in the normal cells or the non-metastatic cells. And this is expression of CTCF. And you can see that the expression level of CTCFL is higher than CTCF in some cells. So this is just showing you a lot of different experiments. And this shows you in each case, 1 is the testis, 2 is the healthy melanocytes, 3 is the primary melanoma cell lines. And these are all metastatic cells, metastatic melanoma in 4 and 5, and the ovarian cancer in 6. So this is CTCF level, and these are the level of expression of all the different isoforms. And you can see that in many cases, the metastatic cells are expressing these isoforms at higher level. So just to summarize what I've told you, there were 23 previously identified isoforms. 17 of these have different coding sequences. 3 are nonsense-mediated decay. 1 has no open reading frame, and 1 we didn't test. So this leaves 13 different coding isoforms. And we were unable to identify 4 of these when we used our panel of cell lines and when we also looked in K562 where these were identified. So we identified 9 of these 13 isoforms by Iso-seq and PCR, and we identify 1 novel isoform from Iso-seq and 1 novel isoform from PCR. So basically 11 isoforms validated. And these are the different protein structures. You can see that these are the 2 novel isoforms, C85 and this is C24. And when you look at all of these isoforms, basically what you can see is that they all have different, either different zinc fingers or different C-terminal regions. And from what I've shown you from the CTCF and CTCFL data, you can appreciate that the zinc, loss of these zinc fingers 4 to 7 is going to make a huge impact on binding and where these proteins bind. And maybe they will act sometimes in competition with CTCF if they have these 4 to 7 zinc fingers. But in other cases, they're unlikely to compete with the CTCF because they don't have the correct zinc finger. So we're now investigating what these do. So just to tell you again, this is summarizing what I've told you, 2 novel CTCFL isoforms, and we identified in total 11 coding isoforms. The expression of each is highly variable depending on the cell type. And overall, the expression level in metastatic melanoma cells is higher than primary melanoma cells. And CTCFL is also expressed at lower levels and sometimes not so low levels in different healthy cell types. And she's now investigating a whole panel of normal cells to see what isoforms are present and are looking at that. So what we're doing now is going on to look at what's the role of each CTCFL isoform and how does it impact CTCF and cohesin binding? How does it impact chromosome structure, gene expression, and chromatin accessibility? And what's the oncogenic impact of each cancer-associated CTCFL isoform? And we're doing this in both vitro and in vivo studies. So basically taking again a function and loss of function approach. So this is the lab. I think I've gone on quite a long time, a bit over time. These are the people that did all the work. We're hiring postdocs if anyone's interested. And these are the funding. And most important, these are the lab people and my collaborators, Tamar Schlick from NYU, Alfej Nora and Benoit Bruno at UCSF, Marcus Noyes, who I should have mentioned before, who's a zinc finger expert who advises me on all of my zinc finger studies, Aris Tsirgus, who's co-mentors a computational postdoc with me, and Jiaodong Cheng from MD Anderson, who's doing all the crystal structures on the mutants at the moment. So I'll stop there and take any questions, and thank you for your attention.
Thank you very much, Jen, for this great lecture. Remember, you can ask questions using the online platform. Just, I'll take the benefit being the organizer. A couple of questions.
Good.
For me. Very general questions. So these different isoforms are generated by different transcription start sites, or you have different alternative splicing, or it's a mixture of both?
It's a mixture of both. Yeah. It's a bit difficult to find where the start site is. I mean, where the alternate promoters are, because we have no way of really accurately doing that at the moment.
And as you mentioned, Boris, the previous Boris, it was described at the beginning only in spermatogenesis, only important at the stage, but you're showing that it's altered in tumors, etc. So this is a question of the variants, no? So the function was established in sperm because only was studied like a main or one isoform, no? I understand.
So no, the guy who did this originally identified CTCFL in sperm was Lobenenkov, and he was the guy who also identified all these isoforms. But going forward, nobody's gone further with that, and people look at CTCFL expression in cancer, they see that it's causing changes, but they're not really identifying which isoforms there. Are there more than one? What are they actually doing? And how do they affect CTCF? So I think it's, and also it's interesting to see that this protein is actually expressed at quite high levels in some normal cells at particular isoforms. So what are they doing? So that's interesting.
And the other is, there are ways to target these proteins using drugs or not? CTCF, CTCFL, there are ways to target the proteins? To upregulate or inhibit them using drugs? Not
that I know of, no. Not so far.
And people have tried to do this induced degradation of the proteins in vivo.
Yeah. They're doing the induced, mostly it's a rapid depletion with oxygen, but we're also doing halo tags now.
Yeah. My final comment, some of the sites of CTCF and CTCFL, there are some methylation dependent.
Yes, yes.
And people, in the past, they did some studies that I remember, no?
Right.
That,
And actually, we are doing some in collaboration with Ben Tycho at the
perfect,
moment.
perfect. Because I think it's very relevant here.
Yeah, and also
in the context of cancer.
yeah, exactly, because methylation levels alter a lot in cancer.
Perfect.
Yeah.
Thank
So
you very
am I
much.
supposed to find questions online here? Or?
Online, I think that they're going to give me
Okay,
a
good.
paper. Or
All right,
something like
I
that.
don't have to deal with that.
Questions?
Hi, Jane. Thanks very much for the talk. So regarding the first part, the mutant, the K2T mutant, you showed that there's reduced cohesin binding, at least by ChIP-seq. So my question is, do you think that that's because CTCF, the mutant, is stopping less cohesin and then it proceeds with loop extrusion? Or is there less cohesin loading on chromatin? Can you check that? And sorry, and related to that, you also showed reduced or altered accessibility of transcription factors in the mutant. How does that work? So how is CTCF binding connected to transcription factor binding?
Okay, I'm going to answer the first part first, which is I think that it's a question of stability and not to do with, I mean, it may have something to do with loading, because I think you affect a lot of genes. So there's a lot of indirect effects. But as a direct effect, I would say it's more about stability. And so we actually are collaborating with Anders Hansen, who does a lot of imaging, and to look at stability by imaging, because he's done a lot of this stuff. So we are giving him all our mutants. So hopefully we'll find something out about that. And the second question, I think, is going to be both direct and indirect effects, because maybe if you don't have a CTCF boundary, you might alter, the gene regulation in some way or other. You might alter the accessibility of enhancers and promoters if they're not, if a promoter is not now contacting an enhancer, or it's contacting a new enhancer that it wasn't contacting before. That will change the accessibility for binding of particular transcription factors. And I'm sure that will end up as being a context-specific thing. So if you look in different cell types, you'll probably find different factors.
Sorry, but you're using to deplete the wild type CTCF, that's a degron, right? So what's the time frame between the degradation and your experiment?
Okay, so we wait 2 days from degradation. And probably we could do it quicker, but this is just, we followed originally when we got these cells from Alphege, that's how he had done them. So we followed what he's doing. But now I think we are starting to do experiments where we do them like at 3 or 4 hours, because I think then you get a much more, a better picture of the direct effects rather than the indirect effects. Both are interesting, because if you're thinking about cancer, you want to think about both things. But if you want to look at the mechanistic stuff, it's nicer to look at the things that happen immediately.
I have a question here. I was just wondering what, if I understood correctly, CTCFL binds stronger than CTCF and displaces it when they're both expressed.
Yes.
So what effect does CTCFL binding and displacement of CTCF have on the gene expression?
Okay, so basically CTCFL is a transcription factor. It's not like CTCF, which can act both as a transcription factor and an insulating agent. But because CTCFL can't interact with cohesin, it can't insulate. So you can have two effects on gene regulation. One is you can lose insulation, right? But mostly the CTCFL binding sites that overlap with CTCF are found in promoters. So maybe it's just acting as a transcription factor in many cases, but because it's kicking off CTCF from a number of sites, you also have this impact on insulation.
So do you see in general that there is an increase of expression of sites where CTCFL has displaced CTCF? I was just wondering whether you have seen this.
If they bind to promoters, yes. Yes, so what's interesting is you get a lot of cancer testis antigens that are coming up. And that is kind of as what you would expect, because this is a transcription factor that's present in testis. And we think that the expression of cancer testis antigens that people see in cancer is all probably related to CTCFL expression. And CTCFL then turns on a lot of other tester-specific genes that are probably important in pluripotency and affecting the cancer as well.
I wanted to ask about the genetics. Do we know if these mutations in CTCF are very early in cancer development, and if they preferentially synergize with some other driver mutations?
Yeah, so we don't have really any information on the patients, which is a pity. And we still don't know the answer about whether these can drive cancer. Well, I've shown you some sort of circumstantial stuff, but these are ES cells, right? So we're putting them into B cells with arimelnic and T cells with Janus, and putting them with notch driver mutations and without, and also with the BCL2 transgene in the lymphoma context. So we don't really know. I mean, what's been shown so far is that if you have CTCF, the heterozygotes, where you've knocked out one of the alleles, they are more predisposed to cancer because levels of CTCF are extremely important. And if you irradiate them, they get cancer much earlier than if you would normally if you would have both alleles present. So we don't really know the answer to that yet. It's a in progress thing.
Hi, thank you for the talk. So I have one question about one of the targets, the DNT3A, I think. So in that case, could you see that the effect of the CTCF mutation affect also the CpG methylation genome-wide?
Yeah, that's something we're currently doing. We just keep adding to our list of experiments. Everything that we find, we add on another 10 experiments. It's becoming quite expensive and a long project.
And just a second, very quick, in the isoforms that you detected, have you also, maybe I missed that, have you also detected the proteins predicted to be coded by
Yes,
those
yeah, I
isoforms?
mean, that's the first thing. Well, so no, we haven't done that in the, we can't do that in the endogenous setting of the cancers because there's no decent antibody that can distinguish between CTCF and CTCFL. You can distinguish between CTCF because you can use an N-terminal antibody and that doesn't bind to CTCFL, but all the CTCFL antibodies we tested, they all pick up CTCF. So you have to do it with a flag tag. And so we're expressing the isoforms in different contexts. And then we, before we do any experiments, we validate with the Western.
Thank you.
Okay. I think for the interest of time, we should finish here. Thank you very much, Jane.
Thank you.